Good evening and welcome. It has been said that history repeats itself. We re-elect the same leaders or members of their families sometimes. We provoke the same crises. We fight the same wars. Yet in science, progress is not cyclical. It's linear and incremental. There's a before and an after scientific discovery, as we have heard in John's talk. The world changed when ether anesthesia was used for the first time to conduct painless surgery right here at MGH in the Ether Dome. There was a world of pain, death, and suffering before penicillin, and a much different world thereafter. Insulin transformed a uniformly fatal disease of childhood into chronic disease in which those afflicted can essentially lead normal lives. And more recently, we have seen the face of AIDS retreat as we win our first round against HIV through highly active antiviral drugs. Well, I think all of us here in this room have lived through one such momentous achievement in human history. Just 10 years ago, we witnessed the complete sequencing of the human genome. To a large extent, thanks to the work of the Whitehead Genome Center, the precursor of the Broad Institute across the river in Cambridge. We now had the building blocks, all the letters, the linear arrangement that decode this language of life into each one of our cells. Just like with ether and insulin and penicillin and HIV drugs, there was a before and an after. The world had changed. There was new knowledge, and there was no going back. And the question we all have was this new information going to transform the face of medicine? Truth is, 10 years out, there is still precious little we understand about this code. There are parts of the genome that we don't know what they code for. There are many genes whose function is still unknown. We lack insight into their mechanisms of regulation. Many of us here and elsewhere are hard at work trying to understand these questions. We want to go beyond a game of Scrabble, of words, a line on a board or in a dictionary, to really compose the unique epic of phrases, sentences, and verses that give us this epic of life. Well, today I want to illustrate how this genomic information might change type 2 diabetes, the landscape of type 2 diabetes, the kind of diabetes that happens usually in adults and typically associated with weight gain. Type 2 diabetes is a modern plague, is a scourge. It threatens to unravel all the substantial gains we have made against cardiovascular disease over the last 50 years. There is the paradox that as humanity marches on to eliminate hunger from the planet, we now have 2/3 of Americans are at least overweight or obese. I just came from Disney World, and I think most of them were there. There's 24 million Americans with type 2 diabetes today, twice the number on the verge of becoming diabetic. And the numbers are only going to double in the next 30 years. The situation is even more dire in China and India and in the developing world. I, as a practicing doctor, I've seen a 41-year-old father of two suffer his first heart attack because of diabetes. I have seen a man in his 30s, an immigrant, first lose one toe, then another, then half his foot, half his leg, and eventually succumb to overwhelming infection in our ICU. I have seen the curtain of blindness descend in middle age. We cannot stand by idle. And not because of these human tragedies, personal human tragedies, but because the explosion of type 2 diabetes and its associated complications of heart disease and stroke and blindness and kidney failure can undermine the whole global economy. We have learned that many of these things happen because of the changes in diet and lifestyle that are typical of the Western world as they spread throughout the world, but they are particularly worse on a background of a genetic predisposition to type 2 diabetes. So in the last 5-10 years, my colleagues and I at the Broad and MGH and elsewhere have identified up to 80 regions of the genome that increase the risk of type 2 diabetes. And in doing so, we have learned of new molecules, pathways, and genes that contribute to the development of the disease and that hopefully will open up avenues for new therapies. Just recently this year, we published a finding by which we know a part of the genome where variation explains up to 20% of the excess of diabetes that occurs in Latino populations when compared to people of European descent. Well, as a practicing physician, I'm most interested in learning how this information might lead to the betterment of my patients. And so we try to understand how genetic information might influence clinical care. We know, for example, that genetic information is not particularly better at improving prediction of who's going to get diabetes than simple factors like your body weight or your blood sugar that your doctor can obtain for you in the office. But we know that genetic information can divide people into high risk and low risk. And that ability to do that enables us to design more efficient clinical trials by targeting to the right population and getting the answer sooner. It also has huge implications for public health strategies. Many people feel that when they have a genetic predisposition to a disease, they're going to get the disease. They're doomed. It's a very fatalistic attitude. Well, we've learned in our own research that an intervention of diet and lifestyle, when deployed in the appropriate population, benefits those at high genetic risk as much as those at low genetic risk. It's equally effective in those groups. In other words, you're not condemned. You're not damned to develop diabetes because of the bad deck of cards that you receive through inheritance, but there's actually something that you can do about it. So another example of how genetic information can lead to therapeutic choices comes in what has been mentioned earlier, and that is pharmacogenetics, or precision medicine, the ability to select the right therapy to the right person based on genetic information. And this has already panned out in rare forms of diabetes, where knowing the kind of mutation a person carries can mean the difference between multiple daily insulin injections or a single pill once a day, or maybe not at all. In our own work, that is partly funded philanthropically through the MGH Research Scholars Program, we're beginning to identify the non-responders to metformin. Metformin is the drug that is used to treat diabetes as a first-line agent throughout the world, and there are some people who don't respond. And we can know that ahead of time. Then the physician, the clinician, can get the right therapy to the right person at the right time. I have to tell you, when you are doing work coming in the morning and you think that what you do may actually transform how medicine is practiced, that is just an exhilarating experience. You really feel like you're standing at a threshold where genetic information can be harnessed for the betterment of humankind. And of all the places and times in the world, really Boston in 2014 is the place to be. Partly because of the major academic hospitals that we have here, the highly ranked universities, large pharmaceutical companies, new and established biotechnology hubs, the insightful venture capital, and all these new companies that arise, all of that come together to really make this a very unique place and attract top talent. And a side benefit is I met my wife at MGH because of the top talent that gets attracted to that sort of thing. But within that constellation of wonderful things that Boston has that is unique in the universe, really, MGH I found to be very, very special. And the reason for that has to do because at MGH we learned to practice team medicine. And team medicine really translates into team science in our scientific quest. So I could speak a lot about what team medicine is on a theoretical basis, but I thought I would just illustrate it with an example. When I arrived here in 1997, my first year of internship, when I'm just learning to be a doctor, we took care of Sam, a man in his 30s. I've learned to call that quite young, in his 30s. And Sam had this cancer that was metastatic that had the particular property that made too much insulin. Insulin is the hormone that lowers your blood sugar. So he had a constant low blood sugar that was not compatible with life. And so he was dependent on an IV that gave him sugar to stay alive. And he was here in the hospital. And we knew, and he knew, that we were losing that battle. Well, Sam and his fiancée decided it was time to get married. He could not leave the hospital, however, so we planned for a hospital wedding in our chapel. There was a fly in the ointment, though. That day, that Thursday, there was a suspicion that maybe Sam had tuberculosis. Tuberculosis is infectious, and so he had to be placed in isolation and had to wear a mask and could not leave the room until we obtained 3 different samples on 3 separate days that were clean of TB. The wedding was that Saturday. So an entire team of residents, interns, nurses, respiratory therapists, laboratory technicians, the chaplaincy, all of us came together with one goal and one goal only. Let's get Sam married in the chapel. So it was very easy that first day with all the excitement to obtain his first sputum specimen. But Friday was rough, and we tried. And the respiratory therapist was working overtime, one-on-one with Sam, until 10 minutes before midnight, we were able to get the second sample. And at 12:01, we got the third, dated on the Saturday. The respiratory technician came from home doing overtime at 5 in the morning to process the samples in time, so that by 11 in the morning we could burst in the room, tear off his mask, and he could kiss his bride and have his wedding in the hospital chapel. So that same team medicine that is quite special about the Department of Medicine in particular here at MGH then translates into our science. The same way that we work together to try to find a loftier goal outside of ourselves for the betterment of someone else, we do in our scientific enterprise as well. We leverage each other's talents. We learn from each other's expertise. We revel in multidisciplinary collaboration. We celebrate each other's insights. And our connections with our sister hospitals, with the Broad Institute, with large pharma, all of that provide a very fertile soil for scientific discovery. When I came to MGH as an intern, as a first time, I had no idea the direction in which my career would take, other than I wanted to work on genetics. And that was in part because my sister has Down syndrome, and I grew up with her, and of course that is a genetic condition. But that's about where it ended. Little did I know that I would be speaking here today, or that my most successful genetic project would be the generation of 4 daughters here at MGH, all born in Blake 14. Well, my family is quite special, not just because of my sister Miriam, but I have an older sister, Toya, who has a different form of intellectual disability. And when she was born, no one could really figure out what the problem was as she developed, and she remained without a diagnosis for 30 years. Now, here I was in 1997. In 2003, we have the human genome sequence. And shortly thereafter, we could tell that there are parts of the genome. I heard a talk right here upstairs in the same case where an invited guest speaker tells us that there is a way to find parts of the genome that are deleted or duplicated in a very, very fine level. Only 2 years later, different talk, same building, a colleague tells me that now it's commercially possible to order the test and get that on a person. So we talked to my sister Toya and said, Toya, there's this test that we can do. It requires a blood draw. Blood draws is something that she hates, by the way. But maybe next time you come from Spain for vacation, we can try to have my friend Joe do this blood draw, and maybe we'll figure out why things are a little more difficult for you than they are for other people. So Toya came, had the blood draw, was quite a trooper, and then went back to Spain. And I will always remember that Friday morning in July when I'm on my computer trying to look up the result, and the result flashes to the screen. It says abnormal. Toya is missing 500 kilobases, a part of the genome in chromosome 17, a syndrome that had never been described before until 2 years prior. And you get 2 references of 2 papers that you can download and then learn about this syndrome. Download the papers. There's pictures of 13 people affected with this syndrome, and they looked like my sister. And the description of the syndrome fit her to a T, including the large nose, which we thought it was a family trait, but actually we were happy to ascribe to the syndrome. Her affable personality, other features were all right there. I immediately called my parents, of course, and the excitement of having that genetic information available and having an answer, especially for my mom, so that she wouldn't feel like there was anything in that pregnancy that had anything to do with the outcome, but really something that we had a name and that we had an answer and a reason. That was enough. That was therapeutic enough and so exciting that they threw a big party, invited the neighbors and all the family, because we now have a diagnosis for my sister 40 years after her birth. So I have seen in my own life that genetics matters, that the genome, the blueprint of humanity we carry all within ourselves, holds many secrets that must be unveiled. In the end, it's anyone's guess whether getting to the answer of whether genetic information will transform medicine is going to be in the positive or in the negative. We don't know. But in 1969, we really did not know whether going to the moon would improve life on this planet. But we had the ability and the technology to do so, and so we went. And just like then, I have the same imperative. The difference is, back then, a lot of what drove us was competition with the Soviets. And what drives us today is really a spirit of collaboration. And I think if all of us, within our different roles, and in society and in humanity, we do our job and we come together to try to bring this forward, within my own professional lifetime, and even in a few years, with type 2 diabetes, we'll unravel the genetic architecture of type 2 diabetes. We'll gain new insights into its development. We will pinpoint new avenues for therapeutic development. We will be able to deploy public health strategies to stem its spread, and we will be able to give people the individual treatment that they need. Thank you very much. Thank you.